Well, Peter, thanks a lot. I really appreciate it. I'm really delighted to be here. I haven't been to the Family Day here. I've been to a couple of Family Days, and I must say this is really absolutely terrific. It's great to see everyone. I'd kind of like to start with a bit of a disclaimer, and then maybe a little bit of a warning. And the disclaimer is that before I started to work at the Foundation, I did basic lab research for about 30 years. And that was working in the laboratory with scientists and going to conferences and presenting data. And one thing you learn about and you work real hard at when you're a scientist is how to present data, mostly using terms which are pretty much incomprehensible to most people. And I mean, if you think about it, the more complicated it sounds, the more important it must be, because if you present things really simply, then people are going to go, oh, that's pretty obvious. So scientists have a tendency to do that. So that's my disclaimer. And the warning is, never invite a basic scientist to give an hour-long talk on a Saturday morning, because this is clearly bad planning, Peter. So I'm gonna start from there and go forward. Peter asked me to talk a little bit about what we're doing now and what we're going to be doing in the future. So that's kind of how I've outlined the talk. I kind of like to start with just a question, which is, when we look here today at 2017, what does the future of CF look like? What are we trying to do at the Foundation? What are we trying to do for all the people with CF, the entire community of CF, from a scientific perspective? Because we've heard about, the breathe cons and the clinical trials and all the things that go into the whole CF community. And I'm going to focus more, I'll bridge a little bit into the clinical trials, but focus more on kind of the scientific aspects of where we are and where we're going. And I'd like to give you a little bit of a context of CFTR modulators. Obviously, they're very exciting, and it's hard not to think about them because of how fast they've progressed. But we're also thinking beyond modulators. And a lot of our efforts, in fact, probably the majority of our efforts, are not really now focused on modulators. How do we expand therapies to people with rare mutations? Are nonsense mutations treatable? And we would think yes. And finally, if we look way out in the future, what do we really want to do for this disease? We don't just want to make more drugs that people take every day and add on to their current regimen, which is already very, very onerous. We want to cure. And there are technologies now that we're working towards to make that happen. We know that the cure is going to be way off in the future. And so we're not just working on a cure. We also think that that's an aspirational goal we want to reach. And our real goal is very simple. We want every person with CF to live a normal life. And there are really four main strategies we have to allow that to happen from a therapeutic perspective. One is organ-preserving therapies. These are also called symptomatic therapies or therapies for treating the complications of CF, and you're all very familiar with those. Those are, pulmozyme and pancreatic enzymes and hypertonic saline. We oftentimes think of them as just treating symptoms, but actually they have a huge impact on lifespan and quality of life. So we are kind of thinking along them more as preserving organ function for as long as possible. Obviously, the CFTR modulators are incredibly important. We want to be able to treat the basic defect, not just the complications of the disease. And we think CFTR modulators will get us a long way toward that goal. The first generation modulators, we're thinking will probably treat about 60%. We'll talk a little more about this in a minute. The second generation modulators, which are now on the cusp of coming into the clinic and being approved, are probably going to reach more than 90% of people with CF. How are we going after rare mutations? This creates a whole new problem. In fact, what's happening in CF is it's going from a rare disease to an ultra-rare disease. Many people with CF have a mutation that only 5 people in the world carry. So how do you develop a drug, a therapy, for a mutation which is extraordinarily rare? We're giving this a lot of thought. This requires a completely new way to think. Nonsense mutations are one class of these mutations, but that means we need different kinds of therapies other than the modulators. We're looking at RNA-directed therapy, and I'll talk about a new term which is kind of creeping into our language, which is something called theratype. And then, as I mentioned, we really want a one-time cure. How do we cure this disease once and for all? We know that the disease is genetic. The disease comes from an abnormality of DNA. So we either need to put back the normal DNA, or we need to repair the mutation in DNA at its source. And that's really where we're going in that direction. So new therapies have clearly transformed CF. There's just no question about that. And progress is rapidly accelerating. You know, the Therapeutic Development Network has performed over 200 trials. Over 25,000 people have enrolled in those trials. We now have 11 specific therapies for CF. They're kind of listed here. The earliest one that was approved specifically for CF, I think, was Pulmozyme in 1993, late 1993. And what these therapies have done is really remarkable, because when we look at the lifespan, the increasing lifespan of people with CF, these are the therapies that have really had that impact. So going from, you know, just a few years old in the 1950s to the teens in the 80s, 70s, and 80s, up into the 30s and 40s and in the 2010s. And as Peter said, huge jump last year, 2016, from 41 years of age to 47 years of age. And we think this will continue to go up. We're only beginning to see the benefits of the modulator therapy. So really, the symptomatic therapies, the organ-sparing therapies have really been responsible. It's better antibiotics, better use of antibiotics, better nutrition, etc. But one thing we don't want to do is go from 11 therapies to 12 therapies to 13 therapies to 15 therapies to 20 therapies so that everyone is now taking more and more and more. At some point, we want to generate therapies where you can start cutting back. And we're not quite ready to do that yet. But we think the modulators could have that impact on certain people, especially young people that can put on modulators. So we're looking at this kind of holistically, and I think there are a number of aspects that, a number of goals that we really want to work with. I'm not going to talk much about these so-called supportive therapies, but I did want to give you just kind of an overview of where we're at. If we look at therapies that treat the complications of CF, like antimicrobials, antibiotics, anti-inflammatory drugs, drugs that will improve mucociliary clearance, nutrition, and lung transplant, we have more programs and more dollars invested in these categories than any other category that we're working on. Because we know that while people are waiting for the new therapies to come out, we want to preserve their function. We want to preserve their lung function, their nutrition, and their health. So these are very, very important areas to us, and I wanted to say that up front because I'm not going to talk much about it during the remainder of the presentation. But we're working on multi-drug resistant Pseudomonas. We're working on Burkholderia sinusopaceae. We're working on non-tuberculous mycobacterium, which we heard about this morning. Really hard to treat infections. If we could come up with a way to thin CF mucus, hydrate the airways, then people without a modulator could benefit from those therapies and have improved lung function and preserved lung function. So this is incredibly important to us, and we have many, many programs. Some of them are listed here. We're working with companies along these lines. We now have, I think, 41 companies right now that have CF-sponsored programs. I'm going to stop here for just a minute and do something which might help if I can. No, I can't. That might keep popping up. Okay, well, we'll just stop. So I think we have 41 programs right now. We're working with drug companies to bring the therapies forward. Not all of those are going to be successful. We know that. But we think that in each of these categories, there are enough drug companies in the market, coming into the market and working towards clinical trials. We will have new therapies in each of these categories in the next several years. I want to start off, though, by talking really about CFTR modulators. Everyone is familiar with modulators. I think it's pretty hard to be in the CF field and not at least have heard about them. So I don't need to really say too much here. But there are really two classes. There's a potentiator, like Ivacaftor or Kalydeco. Then there are correctors, like Lumacaftor or one of the components in Organoids. Potentiators take the protein, which is at the surface of the cell, and they flip a switch. They just turn it on. The protein is sitting there where it's supposed to sit. The problem is it cannot open up, and that's the classic G551D mutation. And what a potentiator does, it goes in, you think it interacts with CFTR directly, or something very close to CFTR, and it just flips the switch. Correctors are more complicated because the protein itself gets stuck inside the cell. It's made inside the cell in a compartment, and it has to move through a series of compartments before it's put on the surface. Many mutations block that whole process of movement. And so the corrector molecules, like lumacaftor, actually interact with that whole process and move the protein to the surface where the potentiator can then flip the switch. And that's why Orkambi has both. It has the corrector to move the protein to the cell surface, and then Ivacaftor to flip the switch. So what has happened? This is kind of a quick update. Most of you are very familiar. The first potentiator was approved in 2012. G551D, 4.5% of people with CF. Landmark for those people because it has a huge impact on their health and their breathing. 2014, just 2 years later, that drug was expanded from the G551D mutation to additional mutations, bringing the number up to 8% of the population. There's a lesson here, and that is when you develop a drug in CF for one mutation, it often works on things you never even thought of, mutations you never even planned that it would work on. That continued to expand. When in 2015, the FDA approved the combination, Orkambi or lumacaftor ivacaftor, which brought the number of mutations or the number of people with mutations that could respond up to about 55%. And obviously, this was for people with 2 copies of F5 weight del. And then earlier this year, 23 additional mutations were added. Again, mutations we never really thought would ever respond. But in the laboratory, people looked to see how they work, and then went into the clinic to test in clinical trials whether or not they had an effect. And so these 23 mutations expanded the population to 58%. But other things happened in 2017. This has really been a watershed year, and I have to say, being at the foundation, it was just amazing to watch every couple of months something would come out. So in March, there was a phase 3 clinical trial report for a drug combination called tezacaftor ivacaftor. And this was developed to be a better tolerated version of Orkambi. Tezacaftor is a lot like lumacaftor in terms of how it works, the level of activity you can get from it. But there's side effects with lumacaftor that cause chest tightness. Some people don't respond to that very well. This phase 3 clinical trial was tremendously positive. The data has been submitted to the FDA, and we expect the FDA will rule favorably, I think, in February of next year. So in just a couple of months, we'll know whether or not there'll be an alternative drug for people with 2 copies of F5 weight del, in addition to Orkambi, which is already available. They also did something a couple months later, which was really unprecedented for the FDA. I mentioned that we're looking at more and more rare mutations, and it's very tough to go into the clinic and test those people with really rare mutations because they're scattered all around the country. You can't get enough of them to get statistical value, statistical significance. So the FDA has been working very hard on this process, and we've had several meetings with the FDA trying to help them understand the dilemma in the CF space, which is we will not be able to do phase 3 clinical trials for a lot of these people with rare mutations. Yet we think we have drugs that they will respond to. And so it's now possible to go into the laboratory, generate the mutation in a cell in the laboratory, and test the drug on the mutation in the laboratory. And the FDA said, you know, there are certain conditions where if you do that, you don't even need to test it in people anymore. You test it in the laboratory under these certain conditions, and we will add that mutation to the label. So this is really phenomenal because it's really never happened before in rare diseases. There's something like it in cancer, although it's very different than what they said for CF. So huge advantage for the whole field. July, just a couple months later, Vertex reported another series of trials. And these, I'll talk about in just a moment, were the phase 1 and 2 trials from what we call triple combinations. This is where you have 2 correctors and a potentiator. So a 3-drug regimen. And these were really unbelievably positive. And they predict that if you have a single copy of F5 weight del, you will benefit from that mutation, which will open up a whole category of individuals that could respond to modulators. And then in August, another 2 months later, the FDA approved Kalydeco for 5 additional mutations, and these are splicing mutations. I'm going to talk a bit about what splicing mutations are in just a minute. So as of now, we're about 60 or a little over 60% of the population that is approved, for mutations are approved, and we think that soon that number will rise even further. So this is kind of what, just a diagrammatic representation of the boxed mutations are mutations which were added to the label in 2017 for partial function. Tripled the number of mutations which can now be treated with this drug under FDA approval. And as I mentioned, this particular trial opened the door to use laboratory data for further extension. These are the 5 additional mutations which are splicing mutations. They have kind of weird names. It's like 3272-26 genes. If you have one of those kind of mutations, it's generally splicing mutations. And so they're very different in terms of their biology, and they're also different in terms of how we're going to develop therapies. So here's where we are now. Lumacaftor, Ivacaftor for about 46% of the population. Ivacaftor in 2016 was 8.3% of the population. This year it was expanded another 4%, so 12.5% to 13% of the population. That leaves a pretty good chunk, untreated people, untreated mutations. But many of these mutations, many of these people with these mutations also carry an F5 weight del. So about 36% of people carry one F5 weight del and one of these other rare mutations. So really important aspect of this from our perspective and for what we think the community really would benefit from, how do we address that 36% of the population? And here's where we get to the next generation of correctors. This has been a project which has really been worked on since probably about 2005. So talking 12 years. We've been trying to figure out better ways to make modulators work on CFTR. And the solution, as we now see it, is 3 drugs, 2 correctors plus a potentiator to really boost that activity. So what do we mean by that? Well, this is a bit of data. I'm a scientist, I'm sorry, I just can't go without a little bit of data. So on here we have the activity of CFTR in the laboratory, cells grown in a dish. And 100% is 2 normal copies of CFTR. And here is where Ivacaftor with G551 gets you, just a little under 50%. And we know that has a huge impact in the clinic. Okay? So if you take a person with 1 copy of F5 weight del, and then another copy of another mutation which doesn't make any protein, class 1 mutation. This is the level of activity you can measure in the dish. Okay? If you give them Tezacaftor, Ivacaftor, this is this new improved version of Organoids, this is where you get about 20%, just under the threshold to make a difference. And that's why Organoids was not approved for people with only 1 copy. It didn't reach the critical threshold. You have to get to about 25% or 30% in order to see that change in FEV1. Okay? But what happens if you take Tezacaftor and Ivacaftor and add a 3rd drug, another corrector molecule? Well, this is data that was presented just last year by Vertex for people with 1 copy of F5 weight del. And the data is even better if you have 2 copies of F5. And what you can see is in the dish, this is as good as Ivacaftor for G551B. And this is what is shown here is a drug called VX152 plus Tezacaftor Ivacaftor, or a drug called VX440 plus Teza-Iva, or a drug called VX659 plus Teza-Iva. 3 different drugs, all added to a dual combination, will get you this level of activity in the lab. So that's pretty exciting. And this is kind of the culmination of about 10 years of work trying to identify molecules which work together to correct a defect. So how does this work? What are we really talking about here? And if this movie works, it might explain things. This is the CFTR protein. It's a model of the CFTR protein. And you can see different pieces of it. So these are kind of the 2 transmembrane segments that are in the cell membrane. These are called nucleotide binding domains. And when you have an F5 weight del mutation, it's slightly abnormal. It's not grossly abnormal, but slightly abnormal. So let's see if magic will happen. So this is kind of a look at the mutant protein. And what we imagine is happening is that 1 drug, the first corrector, goes into one place on the protein, interacts, and stabilizes one part of that protein. A second drug can now come in, because it's still not quite normal. A second drug can come in and stabilize a different part of the protein. Now what you have is a much improved protein that can go to the surface much more effectively. And once it gets to the surface, it can be activated by the potentiator. Each one of these drugs alone does not have much effect. They have little bit of effect. But when you put them together, they work cooperatively. And they have a huge impact on the amount of protein you can put on the surface and the amount of function you can get out of that protein. So we've known this for about 8-10 years. Now what we're seeing is that concept coming to the clinic. So these are the trials that Vertex reported out at this year's North American meeting, which just happened about a week ago. The 440 trial was adding Vertex 440 onto Teza-Iva. That was a 4-week trial. And then the 152 and the 659 trials were 2-week trials. So now comes the big question. Does the laboratory predict what happens in people? That's what makes a difference, because if you have CF, no one really cares what's going on in cell and dish in some lab somewhere around the world. So this is now FEV1 data for people with 1 copy of F5 weight cell. Day 1, study starts. We look at the FEV1. This is VX440. 2 weeks later, 4 weeks later, 12% increase in the FEV1. That is better than what we saw with the early studies of Kalydeco in G551D. So let's look at the next drug, Vertex 152. A good response, but not quite as good. We're at, on the lower dose, 5.6%, and the higher dose, 9.7%. Not quite as good, but still really impressive. Let's look at the third drug, 659. 10% improvement in FEV1. 2 weeks. And you see that improvement the first time you look at it on day 7 or day 8. So this is really, when we saw this data, as scientists and clinical trials people and caregivers, we were literally jumping up and down, because all this time we've been wondering, does all this data in the laboratory work? Will it translate to people? Will it actually have an effect on people? Now this really is validation that the concept in the lab will have a huge impact on people in the clinic. It's still early. It's only phase 1 or phase 2. So it's not yet ready for approval, but it's incredibly, incredibly powerful. So the triple combination results so far are extremely promising. The activity meets or exceeds what we see with Ivacaftor and G551D patients. And you can have 1 or 2 copies of F5 weight cell. If you have 2 copies, you get more activity, but if you have 1 copy, that's enough. Okay? So the data is very consistent across multiple versions of these drugs. 152, 440, and 659 have all been tested in people. Vertex has a 4th drug that they're now starting to bring into clinical trials. And you might ask, why are they doing this with all these compounds? Why don't they just pick one and then move it forward? Well, right now, it's not clear which one's best. It's not clear whether all of these are going to actually survive all the things you need to do in order to get a drug to market. And Vertex is hedging its bets. They're saying, wait a minute, what if 440 doesn't work? Then we have to start all over again with the next one, then start all over again with the next one. By moving them forward simultaneously, they're actually saving time and bringing these drugs to the clinic much faster. They can throw away the ones that aren't as good. They want to take the best ones and move them forward. And when are they going to move them forward? We expect that next year they will start a pivotal phase 3 trial with a triple combination, 1 or 2 triple combinations, using the best molecules they've tested in early clinical studies. And it's not just all about Vertex. So we hear about Vertex because they're clearly in the lead. They made the first drugs. They're ahead of everyone. But there are many other companies coming along with the same kinds of results in the laboratory. Really high levels of activity, combination therapies. Some of these companies are companies like AbbVie Galapagos, Proteostasis, Flatley Labs, and even others. And we're going to start to see those companies come forward in 2018 for their early phase 1 and phase 2 clinical trials to test again, how do they work? Do they have side effects? what kind of level of activity can we get, how much benefit do they provide. And now, what's really great from our perspective is it's kind of a race. May the best horse win. We only hope that we can get the best drugs out there and that at some point in the future, people will have a choice. Well, I went on this triple combination and it kind of worked a little bit, but I kind of want to try the next combination. Using laboratory data, looking at mutations, and trying to figure out how best can we move forward with this. So this is where we kind of think we will be in a couple of years. 2020, 2021, if the Vertex trials work, we will have about 90% of people with highly effective modulator therapy. We also think that there are a number of other rare mutations hiding in the wings, these really rare mutations that will respond to these kinds of drugs. And then there are people that won't respond to those drugs, and I'm going to address that in just a moment. How do we treat them? This is actually really interesting because I'm not exactly sure how it's working. But anyway. So at the plenary, at the first plenary at the North American Conference, this is a statement that Mike Boyle made in front of 5,000 people. And I will say, some people are like, ooh, how confident are we of this? But I think this is actually very true. We will look back at 2017 as the time when highly effective next-generation CFTR modulator therapy went from something that we hoped for. We had the data in the lab, we hoped it would work, to when it became something that we really fully expect to occur. Because now it's just a matter of finding the right drug combination, finding safe molecules, molecules that work really well, and that don't cause side effects, can be taken over a long period of time. And the reason we're confident in this is because there are so many companies moving into the space, so many different approaches, that even if most of them fail, we will still end up with one or two that actually are successful. We're not there yet. There's still a ways to go. We're extremely optimistic. So this is not something you can take to the bank, but if you're a hedge fund investor, this might be something you'd want to put some money into. And I don't say that from a financial standpoint. This is a metaphor. So what about people with rare mutations? And this now has two aspects. One are the rare mutations that could respond to modulators, and one set of people with rare mutations which won't respond to modulators. And just to kind of set the stage here, there are about 1,700 mutations in CFTR, almost 2,000 mutations. There are more than 1,000 mutations less than 5 people in the world carry. Now, looking at the database at Johns Hopkins, and I have to say that Gary Cutting, their geneticist, has really kind of exploded this whole field and gets all the credit for this with his team and all the scientists working with him, has gone through these and is still going through them. And as we stand here today, it looks like there are about 658 mutations which generate a full-length CFTR protein. Those mutations have fairly minor effects on the protein, and we know that some of these will respond to CFTR modulators because we've already seen the FDA expand their label. So how do we now find out which mutations respond to therapy? And that now has brought up a new term we'll probably hear more and more about, called a theratype. And a theratype is simply using laboratory data to predict clinical response. There's nothing new about this. If you think about G551D, it works on Ivacaftor works on G551D. So that's a theratype, or can be. Works on Delta F508. That's a theratype. We now need to extend this process to all the other mutations. And in the past, we've used something called bronchial airway epithelial cells. These are cells that are harvested and taken from lungs, which have come out of people with CF when they go for lung transplant. So if someone with CF goes for a lung transplant, they get a new set of lungs. And rather than take their old set of lungs and throw them in the garbage, we take them and send them to laboratories. They go to laboratories all around the world. There's a couple of them that we really work closely with here in the U.S., and those lungs arrive at midnight, 2 in the morning, 8 a.m. There's a team of scientists in the lab that are assembled the minute they get into the lab, and they start working with them, treating them with antibiotics, treating them with enzymes, loosening the cells from the airway, scraping the cells off the airway, growing those cells, and then freezing them in freezers so they can be used by scientists all around the world. That is really how Orkambi got developed. And that is how all these other drug companies have developed these therapies for the modulators for delta F508. And it's worked really well because delta 508 is a common mutation. And so when you get lungs, it's pretty likely that they're going to have 1 or 2 copies of F508. But it's very unlikely that they're going to have a copy of a mutation that only 5 people will carry. So we can no longer use these primary airway cells that come from lung transplant. So we have to think, how can we do other ways? How can we do other ways? What are the other ways of doing this? One way is to just go into the lab and do it simply. Take a DNA, we're going to code CFTR, put all 658 mutations individually into that DNA. Take all those pieces of DNA and put them in cells that we choose. And we're going to use a tissue rat thyroid and FRT cell. And then with all of those 658 cell lines now expressing all these CFTRs, we just add the drugs. And we say which ones work and which ones don't. And that's kind of similar to what Vertex has done for their development of these extensions for the label. It's also now possible to go into the nose of an individual with CF, make a tiny brush, scrape some cells, grow those in the laboratory, and then test those cells for effect and function on different drugs and CFTR modulators. You can also go into the digestive tract and either do a small rectal snip or into the jejunum through an endoscopy approach and grow those cells. So we now are acquiring a whole battery of ways of testing rare mutations for their effect on modulators. This is the data that we have, which is really instrumental in bringing forward the modulators for F508. This is a graph which shows the change in lung function, the delta FEV1, change in FEV1, versus CFTR activity in these primary airway cells from lung transplant patients in the lab. What you can see is as you get more activity in the lab, you get more activity in the person. And in fact, every dot falls really close to a straight line. And what's really interesting, if you take the mutations, now instead of comparing them to Organoids or Ivacaftor, if you compare them with the triple combinations I just showed, bing, 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 right on the line. So this tells us that the primary airway cells are terrific at predicting what happens in people. And that, when we got the results of the phase 1-2 trials from triple combinations this year, also was incredibly encouraging to everyone that these kinds of laboratory models actually predict what we hope to see in the clinic. This is a bit of a different type of biopsy, a different type of assay. These are cells that were grown from either this, particularly a rectal biopsy, but these can also be taken from other areas of the intestine. You grow them in a particular kind of gel, and they form little balls. And what you have here are F508 del cells grown in what we call matrigel, forming little balls of cells. And when you add something to stimulate CFTR to those cells, for the control cells, which did not receive drug, you see a little bit of swelling. But if you pre-treat the cells, in this case with VX809, which is lumacaftor, and VX770, which is ivacaftor, so this is the equivalent of putting Orkambi on these cells, and then stimulate CFTR, this is what happens. You get a lot more swelling because you have more chloride transport. Chloride brings in water. And so all this water and salt moves into the lumen of this particular organoid, just like it would move into the lumen of the airway and hydrating the airway. So this is work done by Jeff Beekman in the Netherlands. He has a beautiful system worked out, and there's several different people in the Netherlands that are now collaborating with him, as well as in Portugal. And they're finding a very similar kind of relationship. Absolute change in FEV1, lung function, versus organoid swelling. These types of studies can be done on people with any mutation, any combination of mutations. It doesn't have to be common. It can be extraordinarily rare. Take a small sample of tissue, grow it in the lab, test it on a drug, and see whether or not it works. So there's a number of ways that we're using this laboratory data to try to predict which drugs will work and which people will respond. So what about people with rare mutations that do not make CFTR? Well, there's about 800 mutations that have been identified that don't make CFTR protein. These are nonsense, splice variants, deletion, frame shifts. And we know that they won't respond to CFTR modulators because in order to respond to a modulator, you have to have a protein to fix. And about 5% of people in the population carry only these mutations. So a lot of our efforts are now saying, okay, what do we need to do to solve this problem? And to do this, I'm going to just kind of give you a little lesson in biology. And this is pretty straightforward, but it kind of gives a little historic perspective. So we know that the gene, the DNA encodes the mutation. This is the CFTR gene. There's kind of the representation of mutation. DNA is converted into RNA. It's just a copy of the DNA. And if there's a mutation in the DNA, the mutation is now transferred into the RNA. And when you make a protein, that mutation in the RNA makes a little abnormality in the protein. And if you don't form a functional protein, you end up with all the symptoms and complications of CF. So for 50 or more years, this is what we've been doing. We've been treating symptoms. Antibiotics, mucolytics, hypertonic saline, anti-inflammatory, it's like ibuprofen, and obviously a large emphasis on nutrition. Protein repair is CFTR modulation. We're just getting started. We've only had 5 years of experience, and already we've come a great distance in terms of trying to make this abnormal protein look much better. But if you don't make the protein, then you're stuck back here. You have to start thinking about other ways of treating the disease. So can we go in and can we repair the RNA? Can we replace the RNA? Or, even more basic, can we go in and repair the gene defect by using gene editing technology, gene delivery, or stem cells? So I'm going to kind of focus now, the rest of the talk, on what do we need to do for this. How are we going after these problems? Because it's dramatically different than anything we've done up to this point to try to bring modulators to the market. So we started something called a nonsense and rare mutation initiative. Because when you have a difficult problem, the first thing you need to do is study the science. And we started with a funding of 60 new projects. These were academic investigators submitting grants to us where they wanted to study these problems. We're working on this in our own CFFT laboratory in Boston. We got industry involved. We're reaching out to industry. Industry partners are coming to us to say, hey, we have this technology. We have this idea. Could we work together to solve this problem? This is a global effort. It spans everything from really, really basic fundamental studies on protein translation to here's a drug that might work. Let's take it into the clinic. And we've committed more than $60 million to this project to date. So a big focus of ours is nonsense mutations, and this is why. Take a look at common mutations in CF. Well, the most common is F5-Del. In the U.S., this is a couple years old, but about 25,000 people. The next most common mutation has 1,200 people. And that's nonsense mutation, G542X. The next most common mutation has 1,200 people. Then you go down to 7, 6, 6, 5, and pretty quick you're down to 200 or so people. And this is only the top 15 or so mutations. But if you take a look at these, the nonsense mutations jump out at you, because if we could find a therapy for nonsense mutations, then it would have a huge impact in these most common mutations. So I'm going to do something that's a little unusual, and you'll have to bear with me because it's kind of corny. But since I'm a scientist, I'm going to try to make it overly simplified. How many people know what this is? Okay, good. So this tells me something about your musical preferences. This is called cassette tape for the younger generation. And when you take a cassette tape and you put it into a cassette player, you get music. Now, it's pretty hard to explain what a nonsense mutation is, and everyone's heard of them. How many people really, really know how they work? So let's say the cassette tape is DNA or RNA, okay? And you put that into the cassette player, which is actually the ribosome in the cell, and you get music. Now, anyone that has had a cassette tape player in their car has probably been driving down the road at one point or another, and you're listening to your favorite song. Now, in that cassette player, there's a little pin. The pin spins around real fast. And all of a sudden, instead of hearing your song, you hear this, and then it stops. And you know what happened. Because what happened is this. That tape wrapped around the pin got tangled up, and you pull this thing out of your cassette player, and what you get is a string of spaghetti. That's a nonsense mutation. In the middle of the song, it stops. You don't hear the end of the song. Okay? Now, it looks pretty hopeless, but if this has ever happened to you, it's pretty easy with a pencil eraser. See, this is exactly right. With a pencil eraser, you wind that tape backwards back into the cassette, and then you play it again and hope to God that it won't get stuck again and that it will read through the next time. Reading through, playing through that problem. So let's look at this in a slightly different way. Making proteins is like reading. Okay? And DNA is made up of letters. In the case of DNA, there's 4 letters. In the case of English, there's 26 letters. But they kind of can be put around in different ways, and if you put them together, you can make words. Now, in English, there's many letters can make a word, but in DNA, there's only 3 letters. So every word in DNA has 3 letters. Okay? And you can kind of take these letters and put them into sentences. Okay? The big boy hit the red tin can. 3 letters, multiple words, makes a story, makes a lot of sense. And if you put a capital letter at the beginning and a period at the end, you have a sentence and it makes perfect sense. So think of the sentence as a protein made up of letters that are now put together in a very specific way. So what do these mutations do? Well, let's look at a simple mutation where only one letter is changed. This could be E92K, it could be G55D. One letter is changed, and there's the sentence. The big boy hit the res tin can. Now, that kind of makes sense. You can kind of get a sense of what it is, but it's not all the information. Okay? Let's look at delta F508. Instead of changing one letter, you delete 3 letters. And now you have a sentence that says, the big boy hit the tin can. That makes sense, but it's still not quite the same. This is delta F508. You can kind of fix that. You can kind of make it work. But what happens with a nonsense mutation, G542X? Well, instead of having the letters all arranged like they're supposed to arrange, some letters are switched so that instead of having a word, you have a period. And basically, you don't get to the end of the sentence. And now you don't know what the sentence is supposed to say. So it's different to fix a nonsense mutation than it is to fix these point mutations and simple deletions. Frame shifts are kind of an interesting one because if you knock out 3 letters, then all you do is shift to the next word. But what happens if you knock out 2 letters? And remember, a cell can read 3 at a time, always 3 at a time. So this makes perfect sense, but if you knock out 1 or 2 letters, everything frame shifts. So now all the rest of the letters are identical. That makes absolutely no sense. Okay? So we talk about these splicing mutations. Splicing mutations are where just stuff doesn't get processed right. And there's big insertions of stuff that doesn't make sense, or there's big deletions, things that don't make sense. So nonsense, frame shift, splice mutations all have to be dealt with a little bit differently. And in the cell, this all happens by a certain type of protein machinery called the ribosome, and it synthesizes much like the tape player reads the tape. The ribosome starts at one end on a messenger RNA, walks along that messenger RNA, and as it walks along, it shoots this protein out of a little hole, much like the music coming out of the speakers of your player. And as it moves further, the protein grows and grows and grows until it gets to the end. It hits a stop, period, and then the protein is made and everything starts over again. In the case of a nonsense mutation, there's a premature stop. So you never get to the end. The protein is not completely made, and you end up with no CFTR functional protein. So what we're trying to do now is identify small molecules that will go into that stop mutation and trick the ribosome, allow it to read through that stop mutation all the way down to the end and make the full protein. And so we have a number of programs that are working on finding those molecules. There's another problem with these stop mutations because by putting an abnormal stop mutation in the RNA is unstable. It's just degraded very quickly. So we need a second drug, much like kind of the corrector Ivacaftor combination. One drug to stabilize the mRNA and another drug to cause read-through. And we have several programs trying to find both classes of these drugs. Now, this is for Laura Gordon, because she's sitting there saying, this doesn't look very hard. This should be pretty simple. But this is what happens to degrade the RNA. These are the proteins that are involved in interacting with each other, phosphorylating each other, clipping each other, binding to each other, and the end result of all this cascade is that the RNA is degraded. The good news is we're figuring out what these proteins are. We're starting to be able to manipulate them. And there are a number of ways in the laboratory where we can block these pathways. So that's the first step, trying to understand how we might get a drug to do that same thing. So what are we really doing? Well, we're working with many companies. This says 9. I think that number's gone up, trying to identify these small drug-like molecules like Kalydeco and Ivacaftor that will work on these nonsense mutations. We're trying to figure out ways of modifying the RNA, stabilizing the RNA so it doesn't get degraded. We're using something called tRNA, which I haven't talked about, but it's all involved in that whole translation process where we can get these things to go through. Everyone has probably heard about PTC Therapeutics and Adelorn. We had really high hopes. I think we were hoping with all of you that this would work. The drug itself has been approved for Duchenne's muscular dystrophy in Europe, but it has not made it into the U.S. It's unlikely to make it in CF because it simply just doesn't give enough of that read-through. The music isn't loud enough. You can't get enough music to actually hear it. So we went back and we said, let's find other programs that will do this. We set up a $10 million program with Southern Research Institute and the University of Alabama. They've already screened 750,000 compounds. They've identified multiple small molecules that they're working on, and some of them are actually looking pretty promising in the laboratory, and we're continuing to work on this. We have our CFFT lab in Boston, which is working on nonsense mutations, and they have been doing this now for about 3 years, and they've shifted over more than half of their effort to finding, developing, and studying these molecules that work on nonsense mutations. We've also funded a number of academic investigators taking a look at RNA modification. There's things called adenosine deamination, pseudoinylation, cytosine deamination. These are fancy terms, but basically it means it can go into that RNA molecule and switch it from one letter to another letter. And those now are starting to be looked at by companies, saying, wow, this looks pretty good in the research lab. Maybe we can make a drug out of it. Several companies have approached us saying, we would like to take a look at these types of modifications and see if we can develop a molecule that will help this happen on specific mutations in CFTR. Another way to treat this is to just put back the RNA. So if we can restore synthesis of normal protein in affected tissues, we're done. That would be fantastic. We have to get all tissues on this thing. And we know that if you put messenger RNA into a cell, that cell will make the protein. It's kind of like you stick your tape in the player, it plays the song. So that's one way to solve this problem. And interestingly enough, we've had several RNA programs that we're now working with. In 2014, we started a program with a company called Shire, which is a big pharmaceutical company, and they were working on mRNA delivery approaches. Well, in 2016, Shire got sold to Baxalta, or got taken over by Baxalta. And their entire mRNA program got spun off into another company called RNA. So we follow the product. CF programs at RNA. RNA got renamed, and now they're Translate Bio. And a few months ago, Translate Bio came to us and said, hey, you know, we've continued to work on this program you guys started, and we think we got something that looks pretty good. And it's called a lipid nanoparticle. This is another term you might hear, LNP or lipid nanoparticle. And this is where you take an RNA or a DNA molecule and you kind of encapsulate it in a variety of lipids, like fatty molecules. You put a protective coat on it, and you can now deliver this to cells so that when it binds to the surface of the cell, fuses and then releases the RNA molecules inside the cell. And this is kind of a diagrammatic picture of one of these lipid nanoparticles. So they are now far enough along where we are thinking that there will be a clinical trial for RNA delivery, CF, as early as first quarter 2018. So that could be as early as February or March of next year. And we looked at their preliminary data. It looks pretty exciting. There's still some hurdles that need to be dealt with. We want to make sure it's safe. We want to make sure we're going into this the right way, that they're not doing something which would be reckless. But there's another company that we also started a program with called Arcturus. And they have a different secret sauce for their lipid nanoparticle. Just different combination of lipids, different protein, different coat on the surface. And we said, well, wait a minute, this is different. We don't know which one's going to work. We don't know if TransAbio is going to work, Arcturus is going to work. So let's put our money down on several. And Arcturus is kind of working in parallel. They're a bit behind TransAbio, but we just funded $3 million for them to develop their own lipid nanoparticle. ProQR is a company that's trying to do the RNA editing. They started out with Delta F, and now they're looking at other mutations. Ionis Pharmaceuticals, we started a program in 2016 looking for antisense oligos that could go in and stabilize RNA. And we've also now got programs looking at these RNA modification mechanisms. And there, the goal is to change one base. In this case, the stop is the letter UAG. If you could change the UAG to a GG, then the ribosome will read right through and finish making the protein. And this is a way to do this very specifically on CFTR RNA for a specific mutation. So finally, I want to finish on the one-time cure. How can we actually cure this disease once and for all? And there are really two avenues that we're working on. One is gene delivery, which is to either replace CFTR in the affected tissues, or gene editing, which is to repair the CFTR gene. And we're looking at viruses. Non-viral vectors, lipid nanoparticles, can also deliver DNA, and so we're looking at those as well. There are many ways gene editing can work. One way is by CRISPR-Cas9, zinc fingers. Their adeno-associated viruses work in this sense. And one thing we've learned, in order to do gene editing, you have to do gene delivery, because you have to deliver the editing enzymes to the right cells, the right place, in the right tissue. And so a lot of our focus is now looking at gene editing. And we've just inked, I would say, within the last couple months, the first gene delivery program the CF Foundation has funded in about 15 years. And this is with a company called 4D Molecular Therapeutics. They're trying to develop what's called an adeno-associated virus, or AAV. This shows a model of an AAV, and basically what it is a hollow ball of protein. And you can see all these little colored red, blue, and green, and yellow kind of proteins stuck on the surface. And inside is this empty space, which is filled with DNA. So these AAV particles bind to cells, release the DNA into the cell, and then the DNA can go in, make RNA, make protein. And so 4D has a very interesting thing that they've done. They said, well, we want to find an AAV molecule, an AAV virus that's very specific for the lung. So they took all these AAVs and they started shuffling the pieces just randomly. They made 100 million different adeno-associated virus particles by random shuffling of the pieces. And then they screened these AAVs for ones that would specifically bind to lung cells. And they've also looked at heart and liver and other organs as well. And so they approached us with this data, which if you grow these bronchial epithelial cells in the lab and you put this particular AAV on, you can see the green cells light up. That's CFTR, that's not CFTR, that's a marker being expressed in these airway cells. And then they took CFTR and put this in a CF pig and showed that in the pig airway, you could express the CFTR, goes to the surface. It probably doesn't show very well here, but kind of right at the surface of the airway there, a red spot. As the CFTR, it's gone to the surface of the airway in the pig animal. So they're now trying to develop this to bring it into clinical trials in the next couple of years. And there are other programs that are working along the same lines that we're very interested in. Now, obviously, the simplest way to do this is to inhale it. You could take the lipid nanoparticles or the AAV particles, nebulize them, inhale them, let them go into the lungs. For RNA, it looks like it will last probably a week or two, so this might be something you do every couple of weeks. Not quite sure how the dosing will be, but that's where we are right now. For DNA, it could work for months, because as long as the cells are there, as long as they express CFTR, this would be a benefit. But we need to identify where in the lung to deliver them. We need to identify whether it needs to be the surface or the glands. Obviously, we want to make sure that we don't forget about other organs like the GI tract, like the liver, and the pancreas. So there's a long way to go, but we're pretty excited about where we're at. An alternative strategy, instead of putting the gene inside the patient, is to take the gene and put it into cells which are taken from the patient. So if you could harvest cells, grow them in the lab, repair the CFTR gene, and then differentiate those cells into lung cells, put them back into the lung, you could theoretically treat CF in a very, very different way. And what's really pretty amazing about this is people have taken cells, they can do skin biopsy or blood sample, reprogram those cells into something called an induced pluripotent stem cell, edit the CFTR gene in those iPSCs, and differentiate them into lung cells. There have been a couple of labs already that have gotten that far. The trick now, the challenge, is to try to figure out a way to get these cells back into the right tissues of the body. And so we have a number of academic investigators and laboratories and companies that are trying to work on that process. You can also take these cells and grow them up in the lab and actually take normal cells and put these rare mutations in. So you can put 2 X mutations, 2 nonsense mutations in a cell, now grow them up into an airway and use those cells to screen for drugs. And the CFFT lab has already done that for 3 of the X mutations, and they're looking at other rare mutations that we can't get from lung transplants. So very interesting approach with stem cells. How am I doing on time? Can I wind it up here? So we talk about gene editing a lot. You probably have seen this, but I think it's important to kind of look at it again. This is the way gene editing works in the cell, is to go and find the mutation in the DNA and fix it.
In cystic fibrosis, thick, sticky mucus presses down on cilia inside the lungs, preventing the cilia from beating and clearing the mucus. The cause of this problem is an abnormal CFTR protein created from genetic mutations inside the cell nucleus. Our goal is to replace and repair the abnormal DNA sequence to create a functional protein and restore normal mucus. One way to do this is to use gene editing technologies that cut out and replace defective parts of DNA, such as CRISPR-Cas9. The CRISPR-Cas9 repair system has 3 components: a guide RNA that seeks out and binds to a specific location in the DNA, the Cas9 nuclease enzyme, a type of molecular scissors that cuts the genetic sequence at the precise binding site, and a repair template containing the correct genetic sequence for the gene. Once these 3 components are introduced into the cell, the guide RNA binds to and activates the Cas9 nuclease. Now the Cas9 nuclease, the molecular scissors, can search the cell's DNA for potential targets. At a potential target site, like the one shown here, Cas9 unwinds a section of DNA to see if the guide RNA sequence matches. If the DNA sequence matches the guide RNA, the Cas9 nuclease cuts the DNA to create a break in the strands. To replace the mutation with a normal sequence, a repair process is used that precisely edits the DNA. For this editing process to work, a short piece of DNA template that contains the desired normal gene sequence is introduced into the cell. The cell uses this fragment as a template and copies the new sequence as it repairs the broken DNA. Once the first strand has been repaired, the template falls away, and the other strand of DNA uses the repaired strand to fill in the remaining gap, leaving both strands with the corrected gene sequence. The cell then uses the corrected DNA strand to produce a functional CFTR protein that allows the proper flow of salt and fluids through the cell membrane. Mucus returns to normal within the lung, allowing the cilia to beat freely and clear the lungs of germs and irritants.
So that's one strategy for editing. And you can kind of see the things we're thinking about. How do we deliver the Cas9? How do we deliver the scissors? How do we deliver the template? How do we deliver the guide RNA? And all that is now ongoing. If we use that approach, we could treat rare mutations. But we would need to develop a specific correction for each mutation. So if you had G542X or a splice variant deletion, we'd need to develop a guide RNA package. We need to deliver that specific package to each individual person. Another way to do it is, instead of just fixing a small piece of DNA, is to take a larger chunk of the DNA, where you might have 10 or 20 or 50 mutations, and that could be used for a larger number of people. One therapy for a larger number of people, dozens of mutations. What we're really very excited about is something called targeted intron insertion. And this is a way to edit the gene where one therapy would basically be useful for every single person with CF. And this takes advantage of the fact that when the CFTR gene is processed, there are these spacers called introns. And this is the DNA, and the introns are in between things called exons. I'm not going to go into all the detail here, but just suffice it to say that the exons come together and make the RNA. And in the CFTR gene, the problem is we have mutations all over the place. Every single one of these would need to be fixed. But one strategy is to take a normal chunk of DNA as your template and insert it right up at the front end here, so that now you have it in a normal gene, but you have this DNA being used that will be spliced into the right frame. And now you have a normal protein made from a normal RNA, and all the mutation, any mutation you had downstream will be totally ignored. And we think this has the potential to really be of attractive nature to companies that are now looking to go into CF and other diseases. And this is really a large amount of effort trying to get this to work. So I would just summarize here by saying this is an incredibly exciting time. We've never seen things like this happen in CF, both at the clinic and in the laboratory. And our strategy is really to think strategically. What do we want to do now? We clearly want to improve therapies to maintain lung function. So when these new treatments come out, people will be able to benefit from them. We also want to make sure that we finish the task of getting those modulators to everyone, to get them to the 95% of people that can benefit. In the immediate term, we're trying to go after rare mutations with serotyping, nonsense-directed therapies, mRNA therapies. And then in the long term, we want to develop these treatments for the one-time cure. It's going to take time for those. We're not going to wait to get those done. We're working now on all these other things while we're working on the long-term vision. And I'll stop there. And if anyone has any questions, I'll be happy to answer them, or we can talk later at lunch. I'll defer to Peter in terms of how much time I can stick. Okay? Yeah.
So when you were talking, I have two children, well, three kids, but two CFers, homozygous for 508, and two very. So when you're looking at the lung cells versus the CF, are you seeing them respond differently in the lab? Like if you cells from the same person, are you seeing different cells in ways?
Yeah, so the question is, some people, I'm going to paraphrase, but some people have more GI symptoms, some people have more lung symptoms. They have the same mutation CFTR gene. So why would that be? And can you find those differences when you do these laboratory studies? Is that fair? So it's a really great question. We don't know why some people have more GI symptoms and some people have almost no GI symptoms. In the lab, when we look at chloride conductance, they seem to respond pretty similarly. No one has really gone and done a systematic study, though, of patients where you take both samples from the same person and then do a series of people and then try to make that correlation. So I think there's a question. We don't have a great answer for that, but in the studies we've done, which have not been well matched, we've not seen big differences. What we are doing now is collecting nasal cells, blood, and intestinal cells from individual people and banking them at the CFFT lab to do those kinds of experiments. So I think we might get that answer soon. Other questions? This could be good or bad, It could be so clear that there are no questions, or it could have been so obtuse that there are no questions.
Can you just say what the time frame is, you think, for DNA repair, and what time horizon?
Yeah, so the time horizon for DNA repair is very hard to predict. First of all, because that field is moving so fast. Let me start with RNA. If we have a phase 1 trial of RNA delivery next year, and it's positive, and then they go to phase 2, and that's positive, and they go to phase 3, and that's positive, then probably we're looking at 5 to 6 years before it gets approved. So we don't know how long it will be before gene editing can be in a phase 1 trial. It needs to be safe. We need to make sure we don't edit something that we don't want to edit. We need to make sure that we can edit it efficiently. We need to make sure we can edit in the right cells. So we're looking at more than 5 years and probably less than 50 years. but I think 50 years is way too long. We're not going to wait 50 years to get this to work. We're thinking probably 15 years we will have trials, and 20 years maybe we'll have a therapy, and then 25 years it'll become routine. The goal would really be to take these new therapies for cure and really develop them in people that have nothing else. But if they work, and if we can get them to work regularly and efficiently, then all the people that are on modulators don't need to take a pill for the rest of their life. If it's safe and it works well, we could transition everyone from medications for life to a one-time treatment, and then they live a normal life. So that's really our long-term goal. When that happens, that's again spread out a little bit further. But as I said, just 3 weeks ago, a paper came out with an entirely new way to deliver CRISPR-Cas, and no one had ever thought of before. And it worked really well. It got into cells. It used gold beads, and they attached DNA to gold beads and CRISPR to the DNA. It was kind of like this scaffold thing. And I read that paper thinking, wow, this is really cool. We're probably not going to use gold beads, but it really tells you what people are trying to think about in the lab, and we don't know when the next breakthrough is going to come through. So we're watching this field very closely. We have our investigators working on it. The people outside of CF are also going to make big discoveries as well. Yeah. Mm-hmm. No one is. Let me tell you, no one is. It's so new. It's only 45 years old. Yeah. So the biggest concern right now is, and the movie was really neat because, home right in on that spot. It made one clip. Yeah. Yeah. So there are ways that the repair process works, and you can either repair the gene or you can damage it more. So we need to shift this whole process to the repair side of things. And that would be at the site that you're trying to fix. A worry that is in people's minds and has not been addressed yet because it hasn't really gone into the clinic is what happens if a rare event occurs at a site where you don't want it to occur. And so most of the DNA in the body would probably tolerate that, but if it happened to be at a gene that was really important for something, then you could have some side effect from that process. So people are trying to develop these enzymes now and make them more specific, increase the fidelity so that they have very, very low off-target, we call off-target effects. And every couple of months, new enzymes are being ported, which are higher fidelity, more efficient, and also moving more towards this directed repair as opposed to kind of the random damage that can occur. So the jury's not out, but people are working on that, and it's a great question. You know more than you think you know. So a lot of people are trying to figure this out. Yeah.
We haven't really talked about it, but every time a new drug comes out, there's an age when it becomes available to the patient.
So the age issue is above my pay grade because that's an FDA decision, and it's kind of a excuse me, a clinical pathway decision. Probably the most important thing is that it's safe, that you don't make things worse. And we've watched this with Kaleido, starting with adults and moving a little younger, and a little younger. And with cancer, it's a little different because you start with really sick people that have a very short time to live, and you can tolerate a lot of potential problems because there's really no other option, and the consequence is so dire. So CF is kind of at that interface where you have young people that are pretty healthy, and you certainly don't want to go into them and cause problems, which would be worse than disease progression. So I think we will probably always see the sicker individuals starting out for these types of therapies. They obviously can't be too sick, but probably the sicker individuals. And as we get more experience as clinicians and as patients and as caregivers in general, we will then gain more confidence that it's safe, and we'll move it closer and closer to where it will have its biggest effect. Modulators are no different than gene delivery, gene editing. Earlier you can start them, the greater the effect they will have on long-term health and long-term longevity. So I think that it's a process that has to be kind of played out. And you're always going to weigh risks and balances. So the short answer is we don't know, but that's kind of how people are thinking. That's kind of the thought process behind it. We want to get these drugs to people as soon as possible, but you don't want to go back and say, oops, that's something that would be very bad. Anything else? Great questions.
Thank you.
Okay.